Hello everyone. So we continue today with lesson number 5, which is transcription, part of our molecular biology unit. So your note taker looks like this. It's 2 pages. So you can fill out the note taker as we go through the video lesson. So let's begin. Okay, so let's start with, let's start with the central dogma of biology, which is the first part of your note taker. So again, this is a review of what we've seen in the past. DNA can make more copies of itself, and this process is known as replication. DNA can also make RNA. This process is known as transcription. And RNA can make proteins, and that process is known as translation. Collectively, this whole idea of interconnecting processes is known as the central dogma of biology. So let's start with some vocab words. And what is the difference between all of this? So we know that inside a cell, we find a nucleus, and of course only in eukaryotic cells, right? You do have a nucleus. So eukaryotic cells have a nucleus. So what's inside the nucleus? What's inside this brain of a cell? So we know that inside the nucleus we have the genetic material known as DNA. But we also know that DNA can be packaged in different forms, okay? So what are you going to write for definition of a cell? So the cell is the basic unit of life, and inside eukaryotic cells, we find a nucleus with DNA. Now, if I were to take a very compact form of DNA as it's about to replicate, then I would find what is known as a chromosome. And chromosomes kind of look like this, right? Like these little X structures. And we define a chromosome as a single, very long, organized package of DNA, a very compact package of DNA. Humans, we know, have 46 of these chromosomes, one inherited from each parent. And then if we look at particular pieces within the chromosome, we know that these are known as genes, right? So each gene on the chromosome is a particular segment of that DNA. Okay. So let's define a gene or a segment of DNA or a segment within a chromosome. So a gene, these are regions in the DNA that contain the instructions that code for the formation of protein. Humans have approximately 30,000 genes, and each person has 2 copies of its gene, one inherited from each parent. You might remember from the genetics unit, this is what we refer to as an allele, a dominant or a recessive allele. All right, and then we have DNA here at the molecular level. And DNA is your deoxyribose nucleic acid, and these are the instructions for forming species characteristics. Okay, so now that we have the vocabulary all sorted out, let's move on. So you've seen this slide before. These are the 3 main processes that occur in molecular biology. The first process we've already looked at, which is DNA replication that happens in the nucleus. And today we're going to focus on transcription, which is the making RNA inside the nucleus by DNA. The third process is translation, which is making proteins, where RNA makes proteins, and it happens in ribosomes that are free-floating in the cytoplasm of the cell. So all of this inside the cell, right? Now keep in mind that DNA cannot leave the nucleus. It is too big. So it needs to make a messenger, known as messenger RNA, to leave the nucleus, and it leaves the nucleus through the nuclear pores of the nucleus. All right, so for today's lesson, we're going to focus on process number 2, transcription. So what is transcription? Transcription is a process where DNA makes RNA. Where does it happen? It happens inside the nucleus of a eukaryotic cell. When does it happen? It happens during interphase of the cell cycle. What is the enzyme responsible for this process? The enzyme is known as RNA polymerase, and it's the enzyme that unzips the DNA, but also adds the bases for the new RNA transcript that's being made. So mRNA makes a copy of the DNA code for building a protein. So here we see how one template of DNA in pink is being used to make one copy of RNA, which is single-stranded, in blue. So RNA is copying the genetic code from DNA. So what are the three types of RNA that are made? So this is the first one, mRNA, but there's two other types of RNA that are made. There are three types of RNA in total. So mRNA is the first one. mRNA is it's a linear form of RNA. Keep in mind that the nitrogen bases found in RNA are AGCU. Remember, there is no T in RNA, uracil instead of thymine. The other type of RNA that is made is ribosomal RNA, or rRNA. And ribosomal RNA, we're going to learn more about the functions of these types of RNA in our next lesson. But ribosomal RNA is a combination of RNA and ribosomes. And then we have transfer RNA, which looks a little different, and it's a type of RNA that carries an amino acid for protein synthesis. We're going to learn more of this in more detail in our next unit. All right, so what is RNA processing? So when mRNA is made inside the nucleus, it's not really ready to leave. So RNA processing needs to happen before RNA is ready to leave. So when RNA is made by transcription from DNA, it is called a pre-mRNA strand or a primary transcript. This process happens in the nucleus. So this pre-mRNA is just not ready to leave the nucleus with the genetic code. It needs to get processed first. So this only happens in eukaryotic cells where there is actually a nucleus. So notice that the pre-mRNA gets modified or processed in a way. So this pre-mRNA transcript contains segments that are called exons and introns. So here we see that these red segments in the pre-mRNA are kept, right? Are kept in the final mRNA. So these red segments that are kept or left in place are known as exons. They're called exons because they ultimately get expressed. And then we see that these green segments, or introns, they are intervening sequences that do not need to be there. So they're kind of just, messing up the sequence, if you want to think of it that way. So they need to be cut out, and they're actually cut or spliced by an enzyme called spliceosome. So before the final mRNA is ready to leave the nucleus, more of the processing involves adding a 3-poly-A tail, which is a tail of adenine bases, and this is to preserve the mRNA from being degraded, and a 5' cap, which is a little bit the whole biochemical composition of the 5' cap. It's a little bit more complicated, and then you don't need to go into all the detail, but it's just part of the processing to help with the integrity of that final mRNA transcript. So then once it's ready, once it's processed, and you have an unbroken coding sequence of exons, the final mRNA is ready to leave the nucleus, transport itself to the cytoplasm, to translocate within a ribosome and begin protein synthesis. Okay, so now you have a chart here with key components of transcription that we are going to complete together, and let's see if this works here. So it says to label the parts of the lines below and then summarize the steps. So what do we see happening here in this step? Well, we see that the DNA that is in helical formation, it starts to unzip, right? So we know that it's doing that because RNA polymerase is coming in and unzipping it and adding the bases at the same time. So here we see for step 1, the DNA unzips. So this is what you need to write in your note taker, okay? DNA unzips, or if you want to think of it as DNA untwisting. Okay, and then you're going to see that one of the DNA parental strands is going to be acting as a template, and it's actually this one here at the top that is acting as the DNA template for making the mRNA strand. So what's happening here in process number 2? Here we see in gray RNA polymerase, or the enzyme, coming in. Okay, so RNA polymerase comes in. And what does it do? Well, it unzips and it adds the nucleotides, or the nitrogen bases. Remember, the nucleotide is the whole thing: the phosphate, the sugar, and the base. And then what's happening here in process number 3? Well, here we see the mRNA being synthesized or made. So mRNA is made or synthesized. Okay. So what do you see here in number 4? So here is a strand of DNA, right? So this is a DNA template. So don't think of DNA as having a left or right or top or bottom side. So either one of the templates could work as a DNA template for making RNA. So this is DNA template. For number 5, we see that these are the nucleotides, or the bases also in the middle, that are going to attach to the DNA template to make RNA. So these are the nucleotides. Now remember that the nucleotides carry the base, and the base can be A, G, C, or U. No T in RNA. And then here for number 6, we see that this is the new mRNA that is being made. And for number 7, it's pointing to the enzyme that is involved, which is RNA polymerase. Okay. So those are your notes. You can pause this and take notes if you need to. Okay. Continuing. All right, what is gene expression? So if you look at the DNA that is being used as a template to make RNA, there are different sections of DNA. So not all genes within the DNA molecule are actually going to be expressed into proteins, as you see here. So let's read what it says here. Genes used or expressed by the cell may be regulated in different ways. So not all the DNA codes for a protein. Some segments of DNA are involved in regulatory or structural functions, and some have no as yet known function. So we don't even know what some of the genes or segments within DNA are responsible for. So here we see that in this segment of DNA, there is a regulatory gene, and this is a gene that is going to control the turning on and off of other genes that follow the regulatory gene. So this regulatory gene is going to control these promoter and operator genes, which in turn are going to control the structural genes, which in turn are going to control protein synthesis. Okay, so the regulatory genes control the turning on and off of other genes. And then these structural genes here, known as, let's just call them A, B, C, and D, are the ones that are actually going to undergo transcription and these are going to be the ones that are actually going to synthesize and make proteins. So this is an important idea in gene expression, knowing that not every single segment of DNA is going to code for a protein, right? You have regulatory genes that are just there to control, and then you have those structural genes that are there to make proteins. When you take AP or IB Bio, you're going to learn a little bit more about how the promoter and the operator sequences, which make part of these operon sequences, actually do the whole control of protein synthesis, which is interesting to study in the future.